Today, you're currently watching STS with our tagline, "There's been a greater need for accurate and verified information." Today is January 20, 2022, and for today's news, a group of researchers and professionals take place towards a safer community by promoting gene therapy to cure and treat disease. Stay tuned to find out how this could be a life-changing. Scientists and researchers reportedly creating effective discoveries said to be the greatest answer to the underlying problem of disease. According to French Anderson, in the broadest sense, gene therapy is the use of genetic material in the treatment or prevention of disease. The transferred genetic material changes how a single protein or a group of proteins is produced by the cell. Gene therapy can be used to reduce levels of disease-causing version of protein, increase production of disease-fighting proteins, or to produce immune-modified proteins. Cell therapy is a transport or in fact live cells into a patient to lessen or cure a disease. The cells may originate from the patient autologous cells or a donor allogeneic cells. The cells used in cell therapy can be classified by their potential to transform into different cell types. Pluripotent cells can transform into any cell type in the body, and multipotent cells can transform into other cell types, but their repertoire is more limited than that of pluripotent cells. The refrigerated or primary cells are fixed types. The type of cells administered depends on the treatment. Simply, gene therapy works by changing the genetic information of population of the cells in a way that alleviates or combats the cause of symptoms or disease. Here is Naomi Labanyego for the strategies used in gene therapy.
Yes, lovely, thank you. So for the news, gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make a beneficial protein. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. A gene that is inserted directly into a cell usually does not function. Instead, a carrier called a vector is genetically engineered to deliver the gene. Certain viruses are often used as vectors because they can deliver the new gene by infecting the cell. The viruses are modified so they can cause disease when they used in people. Some types of viruses, such as retroviruses, integrate their genetic material, including the new gene, into a chromosome in the human cell. Other viruses, such as adenoviruses, introduce their DNA into the nucleus of the cell, but the DNA is not integrated into a chromosome. The vector can be injected or given intravenously by IV, directly into specific tissue in the body, where it is taken up by individual cells. Alternately, a sample of the patient cells can be removed and exposed to this vector in a laboratory setting. The cells containing the vector are then returned to the patient. If the treatment is successful, the new gene delivered by the vector will make a functioning protein. Researchers must overcome many technical challenges before gene therapy will be a practical approach to treating disease. For example, scientists must find better ways to deliver genes and target them to particular cells. They must also ensure that new genes are precisely controlled by the body. This is Naomi Labanyego reporting.
Thank you, Naomi. So for the uses of gene therapy, here is Charles Darin Manikan for the news.
Yes, lovely. Thank you. For the news, gene therapy attempts to cure sickness and increase your body's capacity to fight disease by replacing a broken gene and adding a new gene. Gene therapy has the potential to affect a broad varieties of ailments, including cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Gene therapy is also known as the gene transfer treatment. The insertion of normal gene into a person's genome to fix a mutation that causes a genetic illness. When a normal gene is injected to the nucleus of the mutant cell, it will most likely to generate into a chromosomal position distinct from the fully allele. While this may repair the mutation, the normal gene integrates into another functioning gene. A new mutation will arise. The normal gene replaces the mutant allele. The transformed cells may multiply and create enough normal gene product for the entire body to be restored to its pre-disease phenotype. Back to you.
Thank you, Charles. Our next reporter is Adrian Lagamo, who is in the Washington, D.C. right now. How are you there, Adrian?
Thank you, Lovely. Yeah, I'm here in the Washington, D.C. It is good to be here and interview the very knowledgeable individuals here in Washington. The news, blood transfusion, as well as the transfusion of red blood cells, white blood cells, and platelets from a donor, is the most frequent method of cell therapy. Another frequent cell treatment that has been used for over 40 years is the transplantation of hematopoietic stem cells to generate bone marrow. Some cell therapies that are available right now are embryonic stem cells. These are pluripotent stem cells that have been generated from embryos. Generally, the embryos utilized to isolate stem cells are used embryos created by assisted reproduction of using in vitro fertilization. Induced pluripotent stem cells. A developed adult or somatic cell such as skin cell is reprogrammed to become pluripotent. These cells have the advantage of pluripotency without the ethical problem associated with embryonic stem cells. Nuclear transfer embryonic stem cells. The nucleus of an adult cell acquired from the patient is transferred to an oocyte or egg cell received from the donor to make these pluripotent cells. Parthenogenic embryonic stem cells. Unfertilized oocytes are the ultimate possibility for getting pluripotent cells. The egg is treated with drugs that promote embryonic genesis without the addition of sperm, and ESCs are extracted from the growing embryo. Mesenchymal stem cells. Mesenchymal are the multipotent cells present in multiple tissues, including umbilical cord, bone marrow, and fat tissue. MSCs give rise to bone, cartilage, muscle, and adipocytes, or fat cells, which promotes marrow adipose tissue. Neural stem cells. Adult neural stem cells are present in small number in defined regions of the mammalian brain. These multipotent cells replenish neurons and supporting cells of the brain. Epithelial stem cells are those that form the surfaces and lining of the body, including the epidermis and the lining of the gastrointestinal tract. Multipotent epithelial stem cells are found in these areas along with unipolar stem cells that only differentiate into one type of cell. Epithelial stem cells have been successfully used to generate the corneal epithelium of the eye. Poietic stem cells are multipotent blood cells that give rise to all types of blood cells. HSCs can be found in adult bone marrow, peripheral blood, and umbilical cord blood. Last, immune cell therapy. Cells that rapidly reproduce in the body such as immune cells, blood cells, or skin cells can usually do so ex vivo given the right conditions. This allows differentiated adult immune cells to be used for cell therapy.
Thank you, Adrian, and keep safe. So for how the virus is used in gene therapy, here is Naomi Labanyega for the news.
Thank you, Lovely. So how the viruses used in gene therapy? As part of their replication cycle, all viruses assault their host and transfer their genetic material into the host cell. This genetic material provides fundamental instructions for making new copies of these viruses. The host cell will follow out these instructions and make new copies of the virus, infecting more and more cells. Certain viruses physically insert their genes into the host DNA. This integrates the virus gene into the host cell's genes throughout the duration of the cell's existence. Viruses like these can be used as carriers to deliver good genes to human cells. First, the scientists will remove the disease-causing gene from the virus. Then, they replace these genes with genes encoding desired effects, such as insulin production in people with diabetes. This procedure must be performed in such a way that the genes that allow the virus to integrate its genome into the host genome remain intact. This is Naomi Labanyego reporting. Lovely?
Thank you, Naomi Labanyego. So for the question of majority, that viral vector can make you sick. There are certain hazards associated with gene therapy. A gene cannot be readily put into your cells. Rather, it must generally be supplied by a carrier known as a vector. Viruses are the most often used gene therapy vectors because they can defect specific cells and deliver genetic material into cells' genes. Some cells get ill because particular genes function wrongly or no longer function at all. Gene therapy is used to fix faulty genes in order to cure an illness or help your body fight disease more effectively. It seems like this generation is more and more productive to help people. Thank you, Naomi, Charles, and Adrian. That was everything about the news for today. I am Lady Jane L. Mariano, reporting.